Well, thank you so much for it's truly a pleasure to have Bruce back on the campus and also to have his family members, and I'll say something about this. This is an exceptional event. A few years ago, starting with our 50th anniversary, we decided we would celebrate on an annual basis at the divisional and departmental level the accomplishments of some of the alumni who have graduated through the department. And so it's my pleasure to collaborate with Bill McGinnis, who is Interim Dean of Biological Sciences. Bill and I have known each other from graduate student days, many, many moons ago. And so it's a wonderful partnership. As I think about what we are doing today and who we are honoring, I want to just relate back to the history of biological sciences. As Pradeep said, this is the #1 ranked biological sciences department in the country, and arguably the first molecular biology department that was established in the United States. So this expands on the theme that this whole campus was set up as an experimental campus. I could name for you a dozen departments that were set up as experimental departments that essentially defined the fields. And this ranking and these accolades for biological sciences would not have been possible without the accomplishments of our undergraduate students or graduate students and postdocs. And so they're the ones on whose shoulders we stand and then receive the accolades. And, Bruce is our first undergraduate student. We've had 3 UCSD alumni who've gone on to win the Nobel Prize. Remarkable thing for me is that all 3 were biological sciences students, alumni. Bruce was the first undergraduate student to get the Nobel Prize in 2011. He got it in the field of physiology or medicine, sharing this with Jules Hoffman and Ralph Steinman in the area of innate immunity and adaptive immunity, and I'll come back to that in a few minutes. He follows in the footsteps of 2 other students who went on to get the Nobel Prize from biological sciences. The first of these was a graduate student, Susumu Tonagawa, who defined how the immune system works in terms of the immunoglobulins and so on. He worked with Masaki Hayashi as a graduate student, almost quit the department, but he came here because it was the first molecular biology department set up in the country. And then we had Venki Ramakrishnan, who got the Nobel Prize in Chemistry in 2009. He came here already, he was a postdoc masquerading as a graduate student. He had already got a PhD in physics, and he came here to learn molecular biology. After 2 years, he quit because he learned enough molecular biology to go on. And I like to say that even our dropouts go on to get the Nobel Prize. Thank you. So this is truly the wonderful accomplishments of our students. I'm also so pleased at a personal level because I happen to know Bruce's dad, Ernest Beutler, who was a distinguished scientist in his own league, and I used to teach the stuff that he used to work on. And today we are truly blessed to have 3 other members, or 2 other members here, but a 3rd member who's not here. All 4 of the siblings in the Beutler family were undergraduates in UCSC's biological sciences. We have Earl and Stephen here today joining us, and so it's just amazing that a family would produce such a distinguished group of individuals, all of whom have gone on to do exceptional things following the footsteps of the parents themselves. We have currently Earl is a businessman and entrepreneur in San Diego, and Stephen specializes in internal medicine and infectious diseases in San Bernardino, California. And Deborah, the sister who's not here, also was a UCSD alum, practices in internal medicine in Pasadena, California. So this fact that this whole family was so immersed in UCSD, and to have several members of the family here today is truly remarkable, and I'm particularly proud of this, and not only in my role as the Executive Vice Chancellor of the campus, but also as a member of the Biological Sciences Department. So on to what Bruce did. At the very early age of 18, he already graduated with a biology degree from UC San Diego. This is the time when most kids are beginning to think of entering college. He graduated in 1976 from Revelle College. And after completing medical school in 1981 at the University of Chicago, he went on to Rockefeller University, then on to UT Southwestern, and then spent close to 11 years right here on the La Jolla Mesa at the Scripps Research Institute. Among his accomplishments, even before he got to the Nobel Prize winning work, he was truly amazing in his scientific accomplishments. He was the first to define a molecule known as tumor necrosis alpha factor and equate it to another molecule that was called cachectin, and showed that the two were one and the same molecule. And the importance of this is that this was one of the molecules that really is a cytokine that is produced by macrophages and plays a very important role in endotoxin-induced shock that is induced by pathogens. And at the same time, though, as this work progressed, he developed a whole bunch of recombinant molecules that counteracted the effect of TNF. And this led in its own right to exceptional work that has led to the production of diseases, drugs that are involved in the treatment of rheumatoid arthritis, Crohn's disease, psoriasis, and this goes back to what Pradeep said about medical applications of basic biological sciences. He then went on to do his pioneering work on defining the innate immunity pathway in mice. And it was known at that time that bacteria, among other things, have a macromolecule called lipopolysaccharides that induce the immune response to pathogens. And he was using a very arduous and tedious pathway, cloned the receptor that is now known as the toll-like receptor 4, and this is a molecule that receives the lipopolysaccharides and responds to it. And interestingly, it turned out that his Nobel Prize winner, the co-winner, Jules Hoffman, had identified a similar type of molecule called the toll receptor in fruit flies, and this is where the two fields converged, one from flies and one from mice. Bruce then went on to use some incredibly amazing and time-consuming and painstaking work using genetics in mice, which is not so easy to do as in flies or bacteria or other systems, to not only develop mice with mutations in the various members of the pathway, but to then slowly and systematically begin to identify all the molecules involved in this innate immunity pathway. And this coupled very nicely with Ralph Steinman's work on the adaptive immune system. And so it's not surprising that many of the things that we talk about in autoimmunity, in cancer, and so on, the discoveries that were made by these three individuals have really transformed the way we think of human health. And it's not so surprising then that the three of them were named in 2011 for the Nobel Prize in Physiology and Medicine. Bruce has received numerous honors, and I could spend the next 10 minutes just reciting them, but I will just draw to your attention the Shaw Prize in 2011. He was elected to the National Academy of Sciences and Institute of Medicine in 2008, the Balzan Prize in 2007, and many, many other honors. He's currently, as you can see here, the Regental Professor and Director for the Center for Genetics of Host Defense, and the Ray and Ellen White Distinguished Chair in Cancer Research at UT Southwestern at the medical center there. So tonight, he will reflect on his experience as a Nobel laureate. He's had a pretty busy year, I can imagine, but most importantly, I'd love to have him share the stories about UC San Diego and how the local community has influenced his life, and I'm sure it's going to be a remarkable story. So please join me in welcoming Bruce.
Well, my thanks to all of you. It's really great to be here among friends and neighbors, and it's a pleasure to talk to you. I was asked to give something of a personal journey to all of you, to tell you a bit about myself, my family, Everyone wants to know something about the person behind the Nobel Prize and about the footsteps that one took to earn a Nobel Prize. So I'll try to do that in a short period of time. And I'd like to start by talking about my immediate ancestors and a little bit about my brothers and sister as well. And with that, I can tell you that I didn't come out of nowhere. I was born to 4 immigrant grandparents, and 2 of them on my mother's side are shown here. Mary Fleischer, born Mary Krasny, and Errol Fleischer, who was known as Harry. I never knew my maternal grandfather because he died before I was born, but I knew my grandmother quite well. I remember her as a very kindly old lady, nearly blind from diabetes. She spent much of the last few years of her life taking care of me when I was a young child. I remember playing games with her, walking around the yard in California. The usual routine was to search for the fountain of youth, which always turned out to be a drinking fountain in the backyard. In any case, she passed away when I was about 8 years old. On my father's side, my grandparents were Käthe Beutler and Alfred Beutler, and both of them were physicians. And while Alfred Beutler, my grandfather, died when I was very young and I didn't know him very well, I did know my grandmother. I was fortunate because she lived to be 102 years old, and I had innumerable conversations with her. She nearly lived in 3 centuries. I asked her, since she was very sharp till the end of her life, what was the greatest technological advance that you remember in your entire lifetime? And I thought surely she would say computers or powered flight or perhaps even the automobile, but it turned out to be central heating was the biggest change to her life because it was always so cold in Berlin. All of my grandparents were refugees from Europe. These 2 came from Germany in 1935 to escape persecution from the Nazis. And my father was born in Milwaukee, Wisconsin, where they settled down. My parents' generation is then depicted here. Probably this would have been taken when they were in their early 30s, shortly after they moved to California. My father went to college at the University of Chicago, as did my mother. That's where they met and married. And my brothers and I were actually born in Chicago. My father was a biomedical researcher, and I'll have a bit more to say about that in a moment because he was a great influence on my life. I was born in Chicago, Illinois on December 29, 1957. And should I ever run for public office, let's say for president, here's the evidence, and you saw it tonight. This is how I looked at nearly a year and a half of age. My first memory was from Chicago. I remember that it snowed probably during the second winter, certainly during the second winter of my life there as we moved to California when I was nearly 2. And my first thought that this must have been something that my brothers did because they were always up to no good, turning the yard white that way. But I didn't see snow again for many years afterward because, as I said, we moved to California in the winter of 1959. And this must have been taken probably about a year after that. This is Steve, who is here tonight, and also Earl standing with me. And I must say, I remember my childhood as being very happy. I remember lots of sunshine, enjoying nature, going on hikes, going to Wilderness Park. I remember there was very heavy smog in those days, which seems to have abated somewhat as I got older. But it was a happy life in California. I grew up in this house. This is the front yard of the house, and this is the backyard of the house. It was quite large, and it had a pool, and it was made of adobe brick, and who could ask for anything nicer than that? So I was not a deprived child. It was really a pretty good life that I had. This picture was taken perhaps 3 or 4 years later. By that time, we had a sister named Debbie. This was taken on a rather somber occasion It was the day of the funeral of my grandmother, my mother's mother, whom I mentioned earlier. She passed away when I was 8, and so I know the exact date of the photograph, and that's why we're all dressed as we are. It wasn't that way every day. Sometime between the ages of 7 and 14, I developed a kind of an obsession with nature and with animals in particular. I was extremely impressed by the fact that they could move, they appeared to think, they had a lot of similarities with humans, and I thought it was absolutely remarkable that non-living matter could be organized in some way along a certain template into living things. And I thought how extraordinary it is that life grabs hold of inert, inanimate matter and co-opts it in that way. I thought surely the name of a science that looked at this must be something along the lines of molecular biology. And one day, my brother Steve brought me a copy of Watson's Molecular Biology of the Gene, and I remember reading it cover to cover with great interest and beginning to understand some of the fundamentals of genetics and biochemistry. I went to a very good high school, to Polytechnic School in Pasadena, and there I learned much more about biology and chemistry and physics. I was very close with the physics teacher in particular, and at one time I thought I might be a physicist, but I realized I just wasn't good enough in mathematics to go very far. It was a time of a lot of intellectual activity for me, I think, a sort of a Renaissance time. I remember from one day to the next, I suddenly fell in love with Bach, and I never lost that love for the rest of my life, but it must have been a maturational thing because certainly I had been exposed to classical music, and yet it just didn't come together for me until I was in my early teens. It was at the age of 14 that I began working in my father's laboratory. And this was a course that I kind of set for myself. It was something that I wanted to do, begged to do, and finally he allowed me to do it. And he set me to work early on characterizing erythrocyte enzymes, also doing starch gel electrophoresis. And I learned too how to fractionate proteins and how to purify them. And I got very, very good at it. This is how my father would have looked at about that age. I take this picture to be somewhere in his mid-40s. And as I said, he was an extraordinary biomedical scientist. He himself had been very precocious. He went to a special program at the University of Chicago, where he began at the age of 15 as an undergraduate and finished with an MD degree at the age of 21. And among his earliest discoveries, he determined that the X chromosome underwent random inactivation in mammals, a discovery also made by Mary Lyon. He discovered it by looking at the erythrocytes of humans, human females, with heterozygosity for an X-linked enzyme, glucose-6-phosphate dehydrogenase. He made many other contributions. He was a great investigator of hemolytic anemias. Also, he concentrated at some times on leukemias and on lipid storage disorders. And toward the latter part of his life, he also did something entirely new. He began to program computers, to write software, to write bibliographic retrieval software, and he ultimately went into business with my brother Earl, who carried on this company for a long time, and it became known as Reference Manager. So I would say he was certainly a polymath, and he was a fine teacher. He was someone who really understood me, and I felt really lucky, not only when I was young, but throughout my life, to be able to talk with him for really most of the last 40 years of his life about science, almost every day. I enrolled at UCSD at a young age, as was mentioned. I was in a hurry. I knew what I wanted to do. I was very focused on biology, and I not only enrolled early, but I made an effort to get through UCSD very quickly. That wasn't for any kind of a bad purpose. It wasn't that I didn't love it here, because I did, but I felt that I wanted to be a part of science instead of really only learning about science. So I began in 74 and I finished in 76. I attended classes, it took a full load through the summer semesters, and I had quite a lot of advanced placement credit, which made that possible. A lot of things were going on in biology in those days. Molecular biology was really coming into its element as a science. People were able, in the latter part of my stay, to cut out pieces of DNA, move them around, express genes in bacteria, and I saw all of that, and it excited me enormously. Genetics was the foundation of all of that, and I realized that at that time. And I was very lucky to have an outstanding genetics teacher, Dan Lindsley, who let me work in his lab for, I think, the better part of a year. There used to be something called a 199, an independent study course, and I took it upon myself to work there, and he was very patient. He let me try to work on something that probably he wasn't interested in at all, which was to isolate hexosaminidase, the Tay-Sachs enzyme from the fly, to try to map the gene for hexosaminidase to a chromosome in the fly. And I learned a lot doing that. I learned about linkage analysis. I learned how to map things, and these were all invaluable to me later on. So you may not have known Dan, but it was an enormous help to me, and thank you very much. By the way, he was kind enough to give me this picture, but I couldn't resist cutting out from the web that picture, which is about what he looked like as I remember it when I was here in the mid-70s. Also, during the summer of 1976, I worked together with Abraham Braudy and more directly with Art Friedlander in his lab. Braudy was one of the pioneers of endotoxin science, and I'm sure it was in that lab that I heard for the first time the word endotoxin and the word lipopolysaccharide. And I came to understand their importance in medicine. They later formed really the core of my work. This was how Abraham Braudy looked around the time I knew him. The question came what to do after college. I did try to emulate my dad quite a lot, and he advised me eagerly to go to medical school rather than to go to graduate school. And the basis of his suggestion was that if I went to medical school, I'd learn all about the workings of an organism. I'd understand about physiology and pathology and pharmacology and anatomy, and I'd be able to put this together and be in a position to do excellent research on whatever I chose to do. He also said if you turn out not to be very good at science, then you'll have a fallback position. You can always be a doctor. And actually, I would say that's probably prudent advice, but it didn't apply to me, and I would never counsel my children that way, because one of the central lessons I've learned in my life is not to prepare for failure. If you start to do that, then you open the door to failing, and you're inclined not really to try as hard, and things don't go as well for you. At least it was always my strategy not to worry too much about not succeeding. So I applied to medical school, and while waiting to hear what would happen, I began working in the laboratory of a friend of my father, Susumu Ono, who was an outstanding mammalian geneticist, also with a more recent interest in immunology. Ono, who passed away in 2000, was known for his discovery that the Barr body, a cytologically observable object in the nucleus of female mammalian cells, was an X chromosome. And that formed a large part of the basis of my father's observation that there was random inactivation of the X chromosome. So it also did for Mary Lyon. Ono was a theoretical biologist. He founded what became known as Ono's law, that an X-linked gene in one mammalian species is X-linked in all mammalian species. And he had, at the time I worked for him, an idea about what major histocompatibility antigens must do. I won't go into this in much detail, but he thought they must be involved in development. And we published a paper on that idea in Cell, of all places. I can tell you today it's completely wrong. There's no truth to that at all. And I didn't really absorb the lesson for a long time, but I'll just say that even very intelligent people, the best theoretical biologists, can often be wrong. And it tended to make me inclined toward a pure genetic approach in my later life. I applied probably to 10 medical schools, and I only got into one of them. I kept getting rejected by one after another, until I was a little bit depressed by it. And I think that might have been because I was so young. And also, I told the interviewers unabashedly that I wasn't really interested in medicine, but in biology. And I don't think most of them took very kindly to that. But the University of Chicago prided itself on training biomedical scientists. And so it was a very good school. And if any place did accept me, if it was the only one, it was a good thing that it was that school. And so this is how I looked. This was my entrance photograph. We were all photographed on the first day of medical school. And I had a lot more hair in those days because my life had not been stressful to that point. Medical school was, in fact, very challenging. I had never worked so hard before I went to medical school. I had never been so sleepless. In those days, it was expected that the junior and senior students had to stay up with the intern, and it was exhausting. And internship and residency, which I did at UT Southwestern, were even more exhausting. I remember a lot of times that I worked 36 hours straight with no rest, no sleep. And this was certainly brutal, particularly as I wasn't interested in clinical medicine at all. But I did certainly learn what my father had promised, that I understood how an organism worked. And also I developed a very strong work ethic. And later, when I began working in the laboratory, I applied the same kind of standards to myself. After 2 years of residency, one in internal medicine and one in neurology, I really badly wanted to go back to pure laboratory research. And I did so, as I'll tell you in a moment. It was during those years also that I became married. I had 3 sons by the year 1987. I got divorced in 1988, and all 3 children stayed close to me, and I took responsibility for raising them in large part, about half the time, or with the older 2, perhaps more than half the time. And that was also extremely difficult for me, given the kind of work that I had to do. I was rescued many times by my administrative assistant, now administrative manager, Betsy Layton, of whom you'll see a picture. But I must just say, every man and every woman should have a Betsy. Between the years 1983 and 1986, I worked at the Rockefeller University in the Cerami lab, first as a postdoc and then as an assistant professor. And the work done there foreshadowed the later work that I would do that ultimately led to the Nobel Prize. It established an excellent marker to follow in asking the question, how does self-non-self discrimination work in the innate immune system? How do our leukocytes, macrophages in particular, recognize microbes when an infection occurs? Because they do that within probably seconds of the time you are inoculated with microbes. I want to tell you about why that question is so important, just briefly. We can say that infection as a phenomenon was appreciated in antiquity by all the great physicians who ever lived. And they knew it by its inflammatory and transmissible character. It could be passed from one person to another. And there were things always in common with infection. Infection would cause reddening or sometimes hemorrhage in the skin if it was in the skin. It would cause all of the things that we know to be associated with infection. But there was no understanding at all, incredible as it seems, that this was something caused by germs. Even after microbes were discovered in the 17th century, nobody made the connection for a long time. They were a curiosity. They weren't known to be the cause of infections. It was Pasteur and Koch who first established that, of course. Koch by isolating microbes in pure culture and passing them back to animals. And the hunt was on for germs responsible for every type of infectious disease during the latter half of the 19th century. There was a student of Koch named Pfeiffer, who's shown standing behind him, who first coined the term endotoxin to explain how microbes were harming the host. He discovered that some structural component of gram-negative bacteria could cause fever, shock, organ injury, all the things that we know a severe infection can cause. Later, it was established that endotoxin was a discrete chemical entity, and so we had something to work with as the initiator of inflammation. And that was very important because we know that severe systemic infections, like in this case meningococcal sepsis, represent systemic inflammation and the collapse of the vascular system as a result. We know also that if we could understand how that came about, what the initiating molecules were, perhaps we'd have a doorway into understanding all inflammation. Because a few years ago, we really couldn't say what lit the spark in sterile inflammatory diseases, like rheumatoid arthritis, for example, or ankylosing spondylitis, or all the other many diseases that end in itis that make up so much of medicine. Immunology as a science suffered from a schism almost from its birth. There were those who studied adaptive immunity, and there were those who studied innate immunity. We think of Paul Ehrlich, the discoverer of antibodies, as the first adaptive immunologist. And we know today how antibodies get to be so enormously diverse. We know from Susumu Tonegawa's work in particular about the genomic rearrangements that create diversity. Elie Metchnikoff, who came from the French school and was affiliated with Pasteur, discovered phagocytosis. And this field was really the founding of innate immunity in a way, although the other side had something to do with it too. But the field languished on the innate immune side, I can say, and it was de-emphasized. Even though every doctor knew that neutrophils were very important and no one can survive long without them, you die of infection if you don't have them, still there was limited understanding as to how microbes are recognized by phagocytic cells and how the innate response is started. It was minimized in most texts. And so this was an underappreciated problem. I came to the field of endotoxin research in a very tangential way. In the laboratory I joined, there was an interest in the phenomenon of wasting in chronic disease. This is something that we see in tuberculosis, in cancer, in many chronic diseases. This is a photograph of a cow that's said to be infected with Trypanosoma brucei. It may have only a few grams of parasite in its whole body, and yet it becomes anorectic and wastes away, and that's one consequence of the immune reaction. It was postulated that the macrophage responding to something made by trypanosomes would produce a protein factor called cachectin, and this would interact with energy storage tissues of the host, like the fat cells, to shut off anabolic enzymes, such as lipoprotein lipase, acetyl-CoA carboxylase, and fatty acid synthetase. And that would lead to the wasting syndrome that was observed. There were variants on the idea. It was thought that tumors might do the same sort of thing, that there might be autonomous production of cachectin from other sources. But as things went along in the laboratory and I began to look at the system, it seemed to me that endotoxin was by far the most powerful inducer of cachectin activity. Furthermore, that the thing to look at was the shutoff of lipoprotein lipase, which was virtually absolute. So I set up a system to purify this factor called cachectin. I did it by growing mouse macrophages in culture, stimulating them with endotoxin, after a period of 2 hours harvesting the medium and assaying the content of cachectin on 3T3L1 cells. I told you I'd had a lot of experience in the past in isolating proteins, even crystallizing one protein, and so I was able to do this quite quickly. And I set up the purification protocol that you see here, and within a few months, I had purified to homogeneity a factor that seemed to be a single protein species that contained all of the cachectin activity, and it amounted to 1 or 2% of the secretory product of the macrophage. It's made in considerable abundance. This is how it looked on a Kumasi stain gel at the time, and I thought it was a unique protein. It was N-terminally blocked. I had to repurify it by another approach to get an N-terminal sequence. In those days, there weren't comprehensive databases of protein sequence, so I didn't know that it actually was similar to something that had been found by others. I sent a sample of this material to Richard Ulevich's lab at Scripps back in about the year 1984, and after a few months, they contacted me by telephone. We used to use the telephone in those days. And they told me, John Matheson called me, and he said, did you know that your protein has very strong tumor necrosis factor activity? It will kill L929 cells in culture at picomolar concentrations. And when I went and looked up the recently published sequence of human TNF, I found there was strong homology. I suspected, and it later became perfectly clear, that what I had isolated was the mouse ortholog of human tumor necrosis factor. Now, this was initially disappointing, but within a few hours, I thought it was more exciting than disappointing. And the reason was as follows. Here, we had one factor, and it was responsible both for killing tumors in vivo or in vitro, and also for shutting off the anabolic activities of fat cells. And both of these were endotoxin-associated phenomena. And I thought, could this one molecule be responsible also for shock, fever, tissue injury, all that we see that goes on when an animal is exposed to endotoxin? I made large amounts of the protein, all from natural sources, and this was quite an effort, but I had enough finally to give microgram amounts to mice. And I found that if you gave 20 micrograms of TNF to a mouse, it would sicken and die, usually, looking very much like an endotoxin-treated animal. And later we found that the pathology in these animals was reminiscent of what you saw in LPS-treated animals. So that was something that was at least consistent with the idea that it was a mediator of shock. More convincing, I raised an antibody against TNF, and I found that you could protect mice against endotoxin challenge by giving them an antibody against TNF. The dose lethality curve would be shifted to the right, and they would often survive a lethal dose of LPS. Though it clearly wasn't the only mediator of endotoxic shock, it clearly was a mediator, and probably the major mediator. Time went on, and many people became interested in TNF, largely for the reasons I've shown you already. The receptors for TNF were cloned in 2 different laboratories by David Wallach and David Goodell at the Weizmann Institute and also at Genentech. And TNF was found to have receptors on practically all somatic cells. Furthermore, whenever it was exposed to cells, it would have inflammatory consequences. In fibroblasts, for example, it would induce the secretion of collagenase, taken to be important pathogenically in arthritis. In the case of neutrophils, it would cause them to express proteins that would make them adhere to endothelial monolayers. And the endothelium would also become sticky. So clearly this was involved in inflammation in general. I thought blocking it might be the most important thing to do in medicine. And I set out to make recombinant molecules that were non-antigenic, stable, absolutely specific for TNF, and that could perhaps be used in therapy if anyone could find the right application. Because it wasn't known yet that rheumatoid arthritis would be so well treated by blocking TNF, for example, and Crohn's disease and psoriasis, as was mentioned. This is how I looked in those days. I was back in Dallas by that time, and here I had made such a molecule. Steve Sprang, who had crystallized TNF and the receptor as well, made this sort of mock-up for me to show, and you can get a little bit better view of it here. What this molecule did was to sequester 2 of the 3 binding sites for the receptor on TNF's surface, and that made it inert and made it harmless, and it would circulate for a long period without activating anything. But this was sort of a digression for me. I was most curious by that time about how TNF was made in the first place. What was the spark that lit the fire? I wanted to find the initial event that occurs during infection. And that became an all-consuming obsession for me. We knew by the year 1990 that there was a part of the LPS sensing apparatus in a molecule called CD14. This was a leucine-rich repeat protein that was anchored to the surface of cells by a GPI tether. So it had no way of signaling across the membrane, and it was widely believed that there must be a co-receptor for LPS that would allow it to signal. The nature of the signal was unclear in large part, But it was known that where TNF was concerned, and most people took that as the relevant endpoint, NF-kappa B activation was crucial. There were 4 NF-kappa B binding motifs in the TNF promoter region, and work with reporter constructs indicated that if you damaged more than 2 of them, then you got no response any longer to LPS. We discovered that once the TNF messenger was made, it still wasn't active, actually. It was sequestered in some way, and a second signal from the receptor would unlock the mRNA, allowing translation of the protein, and ultimately that was processed and secreted as TNF. But the big question was, what's the receptor? This went to the heart of the larger question, how do we recognize microbes? How does the innate immune system become aware of infection? There was a genetic entry point to this question because in the year 1965, it was noticed by chance that certain mice were refractory to LPS. The substrain C3H-HEJ was the first mouse identified in that connection. Unlike all other C3H substrains, from which it had diverged only in the year 1960, you could treat this animal with any amount of LPS and it would not become ill, it would not die of shock. We found that the animals didn't make a TNF response nor any other cytokine response, and that said, probably this was a proximal defect, probably involving the receptor. The same message came from the fact that the defect in these animals was highly specific. They would respond to poly-IC or to flagellin or to lipopeptides, but they wouldn't respond to LPS. So again, there was something very selective going on. Even before anyone knew the nature of the receptor, the mice were used in other laboratories to demonstrate that it was macrophages that delivered the lethal blow when LPS was given to an animal. That was shown by reciprocal bone marrow transplantation studies. Marrow from a resistant animal would make a resistant chimera. Marrow from a sensitive animal would make a sensitive chimera. Oddly, even though the mice weren't endotoxin sensitive per se, they were hyper-susceptible to real infections with gram-negative bacteria. And that taught that the animals must sense endotoxin in order to mount an effective immune response. This was the key pathway that alerted the immune system. It was, in fact, an innate immune mechanism. By 1978, Coutinho and Mayo had found an unrelated strain of mouse with an allelic defect, a mutation in the same gene. This was the C57 black 10 SCCR mouse. You see, they don't look at all similar to C3H-HEJ, but they too were completely resistant to endotoxin. And here too, there was a control strain that was closely related, but was sensitive. By the year 1978, Watson and Riblet had mapped the LPS mutation, as it was then called, to a point on chromosome 4 between visible phenotypic markers MUP1 and polysyndactyly. But there the problem lay for a long time because there were no molecular markers in the mouse, and positional cloning was something that people only dreamed about and had never, ever been accomplished in a mammal. We took up this challenge in 1993. By that time, there were only 300 molecular markers in the mouse across the whole genome. But we thought we might narrow the location of the mutation and we might positionally clone it. And it became an all-consuming obsession of mine that lasted for 5 years. Eventually, we dropped all other work in the laboratory and my entire group was devoted to that one project. These were lean years during which almost no publications came out of the lab. It was kind of a wilderness period, and there were many people who told me just to drop it, but it was hopeless to do. When we looked at mouse chromosome 4, we could find only 11 markers that were informative for a mapping cross that we carried out. Those are the ones shown in circles here, and we didn't really know, because there was no correspondence between visible markers and molecular markers, where the interval of interest might be. So we essentially started over again and mapped the position anew on the chromosome. On 493 meioses, we were able to prove that the mutation was within this green interval between this cluster of 4 markers and this single marker called 80. But that was still a huge area, which might have contained hundreds of genes. And we decided to go further. We did a different mapping cross. We went up to 2,093 meiosis. Every mouse checked for its endotoxin response and genotyped at these markers. And finally, we were able to show with certainty that the mutation had to be between two markers called B and 83.3 that we isolated ourselves from a growing contig in the region. That was an interval size of 2.6 million base pairs. And in those days, we sequenced by hand with radionuclides. And we could, on a good day, maybe capture about 3,000 bases of sequence. And we read it by hand on an X-ray film. And usually I would read and Betsy would type it into a computer. And that was how things went. So that was a huge area to try to explore for genes. Nonetheless, having gone this far, we couldn't give up. And we decided to clone all of the DNA between those two markers into bacterial artificial chromosomes and one yeast artificial chromosome and try to find the gene. Now, in those days, I looked with envy at other positional cloning projects. They usually would involve 2 or 3 backs, and then people would find what they were after. We had a total of 20 backs in our minimum tiling path and 1 yak. We were just very unlucky that this area was refractory to crossing over, and it was even larger than we thought. It's probably about 5 million base pairs in length. We sequenced all of these backs over a period of 3 years to completion, the ones shown in yellow. We were working from the middle outward because the middle was the area of highest probability of finding the mutation. And we had almost completely sequenced everything shown in pink. This was a tedious process where we would fragment each back with ultrasound, clone it into a sequencing vector, sequence in both directions, and then search for genes mostly by a computational method called BLAST. We would blast the sequences against a database of expressed sequence tags. And if we had a strong match, there was a likelihood that there was a real gene in that particular back. So this went on and on and on. And over 3 years, all we found were a bunch of pseudogenes. Not real genes, but close facsimiles of real genes. Only we didn't know they weren't real genes. We had to go to the last nucleotide usually to be quite sure they weren't real genes and to be absolutely sure they didn't have a real mutation. Every one of them looked appealing. We could make up a story about each of them that said this is really the LPS gene that you're after, the response gene. But in the end, all of them had to be discarded. One evening, when almost all of the critical region had been explored, so I was getting very nervous that somebody might have made a mistake, I was sitting at home in my study looking over the day's BLAST results, and I saw a strong hit with a new gene about which very little was known. This was a gene coding for an orphan receptor called toll-like receptor 4. And then I saw another hit a few minutes later, so I knew that this wasn't some kind of mistake. And the matches were almost perfect, indicating that probably this was the real gene and not some homologue and not a pseudogene. I immediately got excited about it because, first of all, the ectodomain of this new gene was leucine-rich, had leucine-rich repeat motifs, and was shaped overall similarly, we would guess, to CD14's ectodomain. As all such proteins tend to make kind of a horseshoe or question mark shape. And it made perfect sense to think, well, maybe the LPS is transferred from one molecule to the next in a kind of a chain, or maybe they come into proximity, and that triggers signaling across the membrane. Another thing that I noticed was that the cytoplasmic domain of TLR4 was very similar to the cytoplasmic domain of the interleukin-1 receptor. Interleukin-1 was an inflammatory cytokine with known inflammatory potential, and its receptor was known very well to activate NF-kappa B. So that made sense too. Here, probably the kind of signals elicited were likely to be the same. Furthermore, I recognized that the namesake of the family, Toll, had been shown by Jules Hoffmann a bit earlier, a couple of years earlier, to be somehow involved in protecting flies against fungal infection. So this was a parallel situation as well. Toll-like receptor 4 might be involved in protecting against gram-negative bacteria, where in the fruit fly, another member of the superfamily was involved in protecting against fungi. Of course, all of this was hand-waving, and one can make up ideas about proteins and feel smug about them, but nothing would mean anything unless we found a mutation both in the HEJ mouse and in the C57 black 10 SCCR mouse. So the very next day, Alexander Poltorak, who was working on this project, dropped everything else, stopped sequencing BACs, and he amplified the cDNA of TLR4 from HEJ and HEN mice by long-range PCR. He then fragmented the whole thing by ultrasound, because that was how we were used to sequencing in depth, and he sequenced it all. And within a couple of days, we found a mutation, the first and only mutation we ever found in HEJ mice that distinguished them from HEN mice. A certain C residue was converted to an A, and at the protein level, that predicted the conversion of a conserved proline amino acid to a histidine. That still wasn't enough, because people might very well say, well, that's just chance. You separate mice for 5 years and look what happens. You get mutations. But that didn't prove that this was the mutation that was causative. So we turned to the other strain of mouse. There we found we couldn't amplify the cDNA. And on a northern blot, we could see none of the messenger RNA. Within a short period of time, we were able to amplify the entire area of the gene in the genomic DNA, and we found that there were 74,000 base pairs missing from that strain. This cut out all 3 exons of toll-like receptor 4 and affected no other genes. So here was an obvious deleterious allele, and the other one very likely was, and without further ado, we published the paper maintaining that this was very likely the endotoxin receptor. We weren't really sure that it was a receptor, of course, and I don't have time to tell you about it, but we adduced very strong genetic evidence that there was physical contact between LPS and TLR4. And that's a complicated story, so I won't go into it, but I'll say that it took 10 years before finally there was real structural confirmation of the receptor-like nature of TLR4, that it really would engage LPS. It had been discovered in the meanwhile that a small molecule called MD2 was still another subunit of the endotoxin receptor complex. This was a basket-shaped secreted protein, and Gio Lee and his group looked at the liganded and unliganded structure of TLR4, and this is what it looks like. It is this beautiful horseshoe-shaped molecule. MD2 fits into the molecule just as shown, kind of occupying a central position. And within MD2, you can see the acyl chains of LPS engaged with this basket-like molecule. They also have direct contact with the backbone of TLR4. And this is where everything starts with endotoxic shock, with this one complex. And in fact, quite remarkably, only about 100 nanograms of this one molecular complex are sufficient to kill a mouse. This will start such a severe inflammatory response that a mouse will die of endotoxic shock if that quantity of receptor is activated. Now, the big excitement for us was that this was one member of a protein family. We know of now 12 mouse toll-like receptors, 10 human toll-like receptors, 13 in both species combined. We hypothesized that each toll-like receptor would have a different specificity. That was a hypothesis that many people had, and it was mainly Shizuo Akira who tested it by knocking out the TLRs one after another. And we know from structural studies now that indeed lipopeptides bind to TLR2, complexed either with TLR1 or 6. Double-stranded RNA is engaged by TLR3. Flagellin binds to TLR5. Nucleic acids like unmethylated DNA bind to TLR9. Profilin binds to TLR11. Collectively, the toll-like receptors can see almost any microbe you might ever encounter And this is where innate immune reactions largely start. Another thing that was amazing was the breadth of utilization of this system in the tree of life. I've told you about the fly. I've told you a little bit about mammals. It is the case that meningococcal septicemia first is initiated via TLR4, and mutations in TLR4 make humans susceptible to meningococcus. But what I really didn't anticipate was that even plants use similar molecules to defend themselves against infection. In the flax plant, shown here, a mutation in a certain motif, tear motif protein that's surrounded by this red box will make the plant susceptible to infection by a fungal rust called Melampsora leani. Now, this is just a part of the molecule that's similar to the toll-like receptor cytoplasmic domain, but the similarity is even greater in other plants, as it turns out. And for that reason, I mentioned this lady here, named Pamela Ronald, who worked on rice plants. She was interested in a certain blight called Xanthomonas oryzae, and she had both susceptible and resistant varieties of rice, XA21 being the resistant variety. And in the early 1990s, she started a positional cloning effort to find out what was different about the two, what conferred resistance. And she came up with a molecule that was called AX21 and XA21. XA21 was the receptor. AX21 was a secreted peptide, a sulfonated peptide made by the bacterium that would activate it. And she worked all of this out with genetics. It was really a tour de force. So you can see we have almost the same kind of molecule in the rice plant, in mouse, and also in the fruit fly that detect infection. Now, evolutionary biologists would like to ask, can we look back and find the last common ancestor? And in this case, the story is almost incredible because we can find the last common ancestor. And the last common ancestor of Pamela Ronald and myself is someone named Fanny Frank. Actually, there were two, also Julius Rothstein. It turned out that she was my third cousin and the very same grandmother who lived to be 102. During the last decade of her life, had been telling both Pam and me, you know, you have a cousin who is working on rice or on mice, respectively. And one of them is at UC Davis, and one of them is at UT Southwestern in Dallas. And neither of us really paid much attention to this. And I guess the moral of the story is that you should pay attention to your grandmother. For the next 13 years, I worked to understand how TLRs would signal. And the last 11 years were spent at TSRI in La Jolla. I was completely enamored of the forward genetic approach by that time. I just loved the power of the approach, the lack of bias, and I saw that we could never have found these receptors without them, understood what they did. My group identified at TSRI about 200 or so, more than 200, new phenotypes in the mice, something like half of them immunological phenotypes in more than 100 genes. And many of them had bearing on how the innate immune system would work, and many of them encoded proteins that at least were not previously known to be associated with immunity. Toll-like receptors began to dominate thinking in immunological circles, and I began to be recognized, although this was quite a gradual process. And you heard about some of the accolades that I had, the Shaw Prize, there was also the Robert Koch Prize, the Balzan Prize. This was a certain prize from the French Academy of Sciences. My boys had grown up by that time, and they came with me to some of these events, and that was extremely rewarding, particularly since I went through kind of rocky times with them when they had been teenagers. But that was nothing to what would follow. And it happened that on October 3rd of 2011, I had just returned from Hong Kong, where there was the celebration of the Shaw Prize, and I was jet-lagged and I was sleepless at about 2:30 in the morning, and I got an email on my cell phone, and I saw the subject line said Nobel Prize. I thought, well, that's kind of interesting. Perhaps this year the Nobel Committee has sent a mass emailing to all members of national academies on whom they rely for their nominations. But I thought I probably at least should open it and not try to go back to sleep. And this was the exact note that I received. It said, "Dear Dr. Beutler, I have good news for you. The Nobel Assembly has today decided to award you the Nobel Prize in Physiology or Medicine for 2011, and so on and so forth. And I was very skeptical. And I thought, how do I really know this is for real? Although I was pretty excited by it. I ran downstairs where my laptop was kept, and I tried to get into the nobelprize.org site, and I couldn't because there was too much traffic. So I went to Google News, and I typed my name. And meantime, I had Betsy on the phone. She had been with me for 25 years by that time. I told her, I think we might have won the Nobel Prize, but let me confirm this. And so I Googled my name. There was nothing. And that was a little bit disappointing. So I refreshed the page, and then suddenly there were 100 news items with the name Bruce Beutler. And I said, it's confirmed. And she immediately started crying. And that began one of the happiest days of my life, I have to say. It was something really extraordinary, and we went to Stockholm together. You can see how it works. You received the Nobel Prize from the king. Your girlfriend is all aglow and looks at your Nobel Prize with you. So does your administrative assistant. Your sons, who formerly viewed you as a nerd, are very proud of you all of a sudden. Your brothers are proud to be photographed with you. Here's Steve. Earl didn't show up for that photograph. I'm sorry. But it was a very happy occasion. And it did change my life. Michael Brown at UT Southwestern told me he thought of his whole life in terms of pre-Nobel and post-Nobel, and it would be the same for me, and it seems to be going that way. One warning I received on the very first day after I met with Al Gilman, probably it was the day after that, was, Bruce, a Nobel Prize is a ticket to travel the world, and if you don't learn to say no, you'll never do anything useful again. And in fact, I heeded that advice. I found that he was right about it. Every day I get 2 or 3 invitations to speak here or there, and I did learn to say no, or I just wouldn't ever be at home. I found that I was called upon to speak with teachers, with students, with donors, with visitors, with various political figures, the president, members of the United States Senate. And this happens quite regularly. I'm also asked to endorse causes I know absolutely nothing about, and I refrain from doing that on all occasions. If I do know about it, I might have something to say. I also thought many people who get the Nobel Prize seem to become smug and complacent and just go around smiling all the time and don't really accomplish any work ever again, and I vowed not to be one of those. But I will say there are lovely things about the prize. You hear from thousands of people. That very morning, I got more than 1,000 emails all at once. And you hear from long-lost friends, and that's delightful. You are treated with more courtesy and respect than you ever received in the past. And family and friends like to bask in the reflected glow of the prize. They're very happy about it. So on balance, I don't regret receiving it. Let me say that. Now, finally, everybody is always asking, to what do you attribute this award? How does one win a Nobel Prize? And I've just told you the whole story, but here are my main conclusions. In my own case, I started out with an earnest love of nature and with curiosity about the phenomenon of life. And I think that must apply to most people who ultimately win the prize. They're interested in nature from an early stage. I think that being a little bit different is helpful. And I attribute the fact that I was a little bit different to my immigrant refugee family background. They had high expectations for the performance of their kids and their grandchildren, and I was eager to please them And it was perhaps for that reason that I went to school early, and I tried very hard to do well academically. I was not so concerned with sports. I didn't really run with the herd, and that's a good thing. I had great luck in my choice of a father, and also I had superb teachers outside my family, some of whom I've talked about already. I didn't prepare for failure as I was instructed to do. I always moved forward with confidence. There have been times I failed, and I haven't told you about that tonight because it's too distasteful even to remember it. But if I do fail, I just try to put it behind me and to move on. I think that I know what's important in science, and to the extent that, that this is a matter of taste, and taste can be acquired, I got that from medical school. And I did choose a problem that was very important, and I knew it was important, but it was undervalued, the problem of how endotoxin works. Once I focused on that, I did it with a relentless obsession. I worked really hard on it, and I didn't work on anything else. Also, there were many people, including my dad at some point, who told me I should diversify or that I should just give that up. It was too hard, and I should work on something else. And I knew it could be achieved. I knew that there was no way that we could fail to find the gene if we were just persistent enough. And so I was not deterred by discouraging advice from other people. And I recommend genetics to anyone. I heartily recommend it to anyone because it was the secret of success in this case. Last of all, one has to pick outstanding colleagues. And this applies, first of all, when you're talking about senior colleagues when you're young. Also, it applies to colleagues who join your group. And I want to say that Alexander Poltorak and also Betsy Layton were absolutely crucial to the success of the whole effort. Both of them worked. Betsy was my admin, worked in the lab as well. And Alexander worked tirelessly, as I did, to try to track down the mutation. Without the two of them, I certainly wouldn't have succeeded. So those are my conclusions, and I want to thank you all very much for welcoming me here tonight, and good night.